From Microbe TV, this is TWiEVO, This Week in Evolution, episode 36, recorded on October 22nd, 2018. I'm Vincent Racaniello, and you are listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Good day from the Elde Lab studios. Good to be with you.
Time of the month, Nels.
Yeah, we're back at it. Episode 36, 3 dozen, if my math is correct.
Yeah. Everything well with you?
Things are great. Gorgeous autumn early afternoon here, although some storm clouds on the horizon. Looks like we might get a thunderstorm this afternoon, which would be great. Start collecting some snow at the higher elevations.
All right.
How are you?
I'm pretty well.
That sounded almost convincing.
Yeah, I'm okay. I could be better. Yeah. I am alive and healthy, though, so I think that's all that matters.
I'm with you there.
But the one thing I have to say is that I continue to think about cool science, and so that's what we're all about, right?
Yeah, now we're talking. Speaking of that, I think we should jump right in and introduce our guest today on TWiEVO.
Why don't you do that, Nels?
Yeah, so this is really fun. A great colleague, Mia Levine, who runs her lab in the Department of Biology at Penn, is here with us. She just posted, I think in the last week, a really spicy new manuscript on the bioRxiv that we'll be talking about today. So welcome, Mia.
Thanks for having me. I'm happy to be here.
So, Mia, do you recognize spicy as a Nelsism?
That is exactly what I was thinking as he said the word spicy. That was generally used to refer to genes evolving under positive selection through using PAML, but I see that it's applicable to other instances as well, which is great.
Yeah. I've heard it many times.
Well, we should probably say, so Mia and I overlapped as postdocs at the Fred Hutch in Harmeet Mullick's lab a few years back.
That's right.
And did you leave the lab before Mia did, Nels?
I did. So I went on in 2011, started my lab in July 2011, so now a little north of 7 years, actually. And Mia, when did you start yours? 2015? Is that about right?
I did start mine in 2015. I'm just entering my 4th year.
Mm-hmm. Just for comparison, a little bit of relativity, I started my lab in 1982. I think before... were you guys born yet?
Yes.
Probably you were, but you were very young. You were very young. Mia, where are you from originally?
I'm from Montreal originally, but I grew up in Los Angeles.
So you would consider LA home?
I would consider LA home, yes.
Yeah.
But I was actually an undergraduate at the University of Pennsylvania. So coming back here really does feel like coming home, possibly more than Los Angeles does.
What did you major in at Penn?
I majored in biology. I am down the hall from several of my professors who taught Bio 1 and 102 to me and very much inspired me. And so it's really a trip to be able to say hello to them every morning as I walk to my office.
So at that time, were you thinking of a career in science or something else?
I was very much thinking of a career in science. I showed some aptitude when I was young, and my father's a physician, and so it was quite natural to presume that if I was good at science, I would pursue medicine. But it was actually in the class of one of my current colleagues that really drove home the possibility that I could do something else with science. I also have an older brother who is a scientist, and he really kind of paved the way with respect to my family to make science a real possible career option for me.
Cool. What did you do after Penn?
After Penn, I went down to the Smithsonian and I did some ecological research and conservation outreach on mangroves in Belize. I then went to the University of Illinois, where I pursued plant ecology And it was during that time that I decided that evolutionary timescales were more interesting to me than ecological timescales. And that led me to seek a PhD somewhere else. That took me to the University of California at Davis, where I did work on molecular population genetics of Drosophila.
So that's the origin of Drosophila that we'll talk about today.
That's right.
How'd you like Davis?
Davis was phenomenal. It was a super place to be a graduate student. I was very lucky. Pursuing molecular population genetics at Davis was a real privilege. There was sort of a pantheon of population geneticists there, including John Gillespie, Chuck Langley, Michael Torelli, and my own PhD advisor, David Begun. And so from them, as well as Sergei Nuzhin and Artyom Kopp, it just was a tremendous place to think about Drosophila evolution, and has obviously had a lasting impact on my career now.
Yeah, great place. And just to echo on that a little bit, now Graham Coop as well has kind of joined that crew.
That's right. That's right.
Really good spot for population genetics, and continues into the next generation.
So we heard that you went to the Malik Lab. What'd you do there?
Well, Harmeet's lab was really a dream for me, because I worked on the evolution of young genes in Dave Begun's lab, specifically the evolution of de novo genes. So I was already thinking about gene birth and gene death. And then I also got interested in chromatin proteins, and I spent a lot of my PhD looking at the evolution of chromatin factors, specifically in adaptation to novel habitats. And towards the end of my PhD, which was really, DNA sequence analysis primarily, I became quite frustrated with the fact that I wasn't looking at the evolution of function. And so Harmeet's lab really brought together those two independent interests, really, during my dissertation research into one lab. And I had the privilege to adopt an entirely new toolkit to complement the one I developed as a PhD student, to look both at the evolutionary genetics and genomics of chromatin, as well as thinking about the sort of cell biological consequences of their evolution.
So while you were there, did you work on telomeres at all?
I didn't work on telomeres, although it was part of my K99 proposal. So at the very end, as I was thinking about my transition, I started exploring that part of the chromosome.
What was attractive to Penn for you? I'm sure you looked at a couple of places.
I did, yeah. Sort of like Harmeet's lab, bringing together kind of independent interests into one place. Penn is this tremendous place to do interdisciplinary work in evolution and sort of chromatin biology and epigenetics. So my department of biology is tremendous for breadth and depth in evolution. We have people writing down equations, we have people in the field, we have people working in cell culture, thinking about evolutionary questions. And then at the same time, at the Perelman School of Medicine, I am able to interact with a group of extraordinary chromatin biologists through the Epigenetics Institute, headed by Shelley Berger. And so with this combination, I really have an extraordinary opportunity to kind of bring together my independent interests in evolution and chromatin biology. So it's really a dynamite combination.
I love the name of that institute, the Epigenetics Institute. I don't think I've ever heard of an institute with that name. It's really cool.
Yeah.
Do you know Sarah Tishkoff?
Of course. Sarah is in our department, as well as the Department of Genetics.
Cool.
She's a fantastic colleague. And as we start to think about moving, not moving out of Drosophila, but complementing our work from Drosophila with human genomes, I think she's going to be a really important piece of that extension of our work.
Yeah, she was a guest on TWiEVO not too long ago. A really fun interview.
Yeah.
She's really an inspiring scientist.
Yeah, 28. That was back in February. And we talked about the genetics of skin pigmentation in Africa. That was cool.
That was fun.
Fantastic paper last year.
Well, last night or a couple of nights ago, Nels emailed me a link to your new bioRxiv contribution. So why don't we jump into that?
Okay.
Yeah, so the diversification and collapse of a telomere elongation mechanism. So Mia, when I was growing up as a scientist, the telomeres...
I don't know why I'm laughing. Sorry.
Me either. Come on, Vincent, I worked on that line for so long. No, but we all kind of learned, I think, from the textbooks that the ends of the chromosomes, the so-called telomeres, are pretty well-behaved parts of the genome. So that there is this problem, the so-called end replication problem. Maybe you can help us define that a little bit. But that had been solved by an enzyme called telomerase, and it seems like things are pretty kind of organized and calm out there at the chromosome end. But maybe give us a little background on the situation in real life.
I think it's attributed to Watson, this end replication problem, which really refers to the nature of DNA replication at the very ends of our chromosomes. So if you remember back to genetics, you might recall that the leading strand is synthesized continuously and seamlessly, while the lagging strand is synthesized in these short Okazaki fragments. And this arises because DNA polymerase moves only from the 5' to 3' direction. So at the very end of the chromosome, the leading strand is synthesized completely, where the lagging strand relies on short RNA primers from which these Okazaki fragments are generated. The RNA primers are ultimately degraded. Polymerase comes in and fills in the gaps between these fragments. But this then leaves a gap at the end of the chromosome, and if left sort of unaddressed, each cell cycle, DNA will be lost. And so virtually all eukaryotes rely on telomerase to bring a sort of repeat element to the ends of chromosomes, which is reverse transcribed from RNA into DNA. And then the RNA primer can then be built on this repetitive DNA, which can also be degraded without any deleterious consequences. And this preserves the unique sequence, our genes and regulatory information that are proximal to what we call the telomere.
Mm-hmm. So to remind us, so that repetitive sequence out at the ends of many eukaryotic chromosomes, it's that like a 7 base pair repeat, or what is?
Yeah, hexanucleotide repeat. It varies depending on the eukaryote, but for our purposes, it's this hexanucleotide repeat, and it changes very slowly over evolutionary time, as you might expect from something that is sort of doing a good job at a really important job for the genome.
Mm-hmm. Yeah, and so I think we all probably heard about telomerase from our science textbooks. And in fact, that was, the Nobel Prize, I think, in 2009. This was Elizabeth Blackburn, Carol Greider, and Jack Shostak. And, one of the reasons I really kind of loved this example was so Greider and Blackburn were working with the model ciliate tetrahymena, which breaks its, when it undergoes sort of nuclear development from the germline to the somatic nucleus. So these critters have 2 nuclei. They're pretty complicated for a single cell. They fracture their chromosomes into hundreds of kind of micro chromosomes and then amplify them. And so there you have not only hundreds of chromosomes, but 2 times linear chromosome ends.
Right.
And so you need a lot of enzyme, you need a lot of telomerase to solve that end replication problem. And so using biochemistry, they were able to purify that activity from tetrahymena, one of the sort of, I think, really beautiful examples of using the right genetic model system for the problem or the question that you were asking. Yeah.
I was just going to say that. Absolutely. A brochure for model organism research.
So telomerase is very old in evolutionary terms, right?
It is very old, yes.
I think yeast have telomerase, correct?
Yeah. They do.
And this is a problem when we get old, right? Our telomerase loses activity and our chromosome ends shorten. Is that the idea?
So it's my understanding that telomerase is most active in our germline, and so our somatic cells actually don't express telomerase. And so the problem of aging arises because telomerase is not sort of reactivated as we get older and our telomeres shorten. Although this exception, is a really important exception, of course, is in cancer, where telomerase is reactivated, except actually in 10 to 15% of cancers, which we think might be somewhat relevant to our discoveries in this preprint, which is that these cancers do not reactivate telomerase, despite the fact that the cells are dividing and dividing, but instead rely on an alternative mechanism of telomere elongation, which is a recombination-based mechanism. And you can sort of, in simple terms, you can imagine the shorter telomere copying its sequence off of the longer telomere. And we wonder, in fact, if this sort of alt pathway, as it's called, and is sort of slowly being worked out in human cancers, might actually be relevant to one of the species that we uncovered having unusual telomeres in Drosophila.
That seems like a pretty straightforward mechanism, and you wouldn't need a telomerase, right?
Yeah, I hope the reviewers agree with that.
Well, that brings up the question of why we don't do that instead of use telomerase, but that's kind of an unanswerable question, I suppose.
Yeah, I think that it's more error prone than telomerase.
I see.
But,
Yeah.
So telomerase, I mean, in some ways seems like a great solution to this issue, but
Absolutely.
it's not so simple for some other species. So you mentioned, I think in the intro of your preprint, that some species of fungi, algae, some moths, crustaceans, and then the topic that you really dug in on, the diptera, or the flies, in this case Drosophila, that something else is going on. So tell us a bit about that.
Yeah, so there's an accumulating list of eukaryotes that do encode telomerase, but they also appear to have insertions of a particular class of transposable element called the non-LTR, or long terminal repeat retrotransposons, that move around using reverse transcription, which sort of sounds a lot like telomerase in some ways. And these telomeres may have that hexanucleotide repeat, but they also appear to have these insertions. And in the moth, for example, in Bombyx mori, telomerase expression is pretty low. And there's a lot of these retrotransposons that live at the ends of moth chromosomes. And we think of Drosophila in this context as the most extreme case, where telomerase has been completely lost and no such hexanucleotide repeat is there. And instead, melanogaster chromosomes are populated entirely by this non-LTR retrotransposon class, and specifically a subfamily within that class called the jockey elements. And these elements are found throughout the genome typically, but this subclade within the jockey family is associated with inserting exclusively at telomere ends in Drosophila. And that's how we really entered this problem. It's really quite tempting to sort of speculate that these retrotransposons are a functional replacement for telomerase. I mean, they use reverse transcription. They're only found at the ends of the chromosome, so they seem to obediently maybe avoid jumping anywhere else where they could be hazardous. Somehow the telomeres of melanogaster don't have any other retrotransposon or DNA transposon at their chromosome ends, suggesting possibly that the host genome has some mechanism of keeping out sort of interlopers. And so, not surprisingly, the discoveries associated with the Drosophila telomere, really, I should point out, have been pioneered by Mary Lou Pardue, Jim Mason, Harold Beesman, various others, suggested that there was this really nice functional replacement and has been referred to as a sort of genomic symbiosis, a kind of cloudfish sea anemone relationship between the host and its retrotransposons, where the host is providing this hospitable niche at the end of the chromosome, and the transposable elements are sort of reciprocating by providing this important genome service so the host doesn't lose important unique sequence. And go ahead.
Oh, just going to say, it's really wild that you would sort of turn over the keys to your chromosomes to these sort of retrotransposons. And is that right? So it's different in moths where they still have some remnant of telomerase activity,
yes,
but in the flies, it's been completely lost.
That's right. And we know that loss event now is actually quite ancient. Although I should say that, few diptera outside of flies, mosquitoes, and some midges have been studied with respect to their telomeric sequence. But the telomerase loss appears to be quite ancient. And it was really only in putting together this paper that we began to appreciate that Drosophila might be quite unusual for having both lost telomerase and replaced it completely with these retrotransposons. But that maybe is me jumping ahead.
Can I ask you a quick question? You mentioned earlier how telomerase is really done at birth, and then you're stuck with the number of repeats you have. Is that the same with Drosophila? Are the flies born with a certain number of TEs, and in the lifetime of the flies, there's no more transposition, or does it happen actively? Do we know?
That's a great question, and the answer is that we don't know. The sort of somatic transposition rates are really poorly characterized in Drosophila, but I think it's a really interesting question. And I like that question a lot because we think a lot of this dynamic evolution is being driven by the germline processes where transposable elements have access to the next generation. But if the host genome is evolving as we think it is based on our findings, is evolving to really police these elements, then it suggests that this telomere isn't particularly maybe optimized. It's more of a negotiated truce between these two parties, and maybe there are some sort of consequences for somatic telomere maintenance. But again, that's for a sort of future study.
Got it.
So you started with this idea, or the history of the idea, that this is sort of a symbiotic scenario. The transposable elements sort of have a home in the genome, and the host's happy because it solves the end replication problem. But now it sounds a little more like a truce or a conflict. So maybe take us into how you attacked that idea with the preprint.
So we're interested in the possibility that these retrotransposons are evolving to escape this maybe a jail at the end of the chromosome. Maybe they're happy to colonize that space, but the sort of null hypothesis, if you will, for retrotransposons would be that they're evolving to increase their own copy number. And so surely being offered this residence is insufficient. And so we would expect possibly that the retrotransposons are evolving to maybe lengthen chromosomes well beyond what the host genome needs for ensuring unique sequence is preserved. Or alternatively, they're evolving to kind of escape this jail and get into other parts of the genome. No one really could explain to me why these retrotransposons don't also live, for example, in the pericentromeric heterochromatin, where they're not at risk of disrupting genes. So if indeed these retrotransposons are evolving or sort of innovating to kind of escape host control, we would predict that the elements themselves would be quite dynamic over evolutionary time. And so we first, really with quite modest goals. In fact, this was supposed to be sort of figure 4 of a paper we published 2 years ago, sort of checking off the list the idea that these retrotransposons are really dynamic over evolutionary time. And we sort of got into this, and it became more and more interesting, and decided to really explore it more thoroughly, which is what you found in the preprint. And so we looked to this melanogaster species group. The names are not really important. What is important to know is that melanogaster and its close relatives have actually been pretty well studied for these retrotransposons that are specialized at the telomere. And these close relatives all encode the same lineages. In 2007, the late Alfredo Villasante, Jose Habad, and colleagues looked across what was called the 12 Drosophila genomes, which captured about 40 million years of evolution, and discovered that there were other retrotransposons that appeared to be related to this jockey subclade, but in fact, were not sort of one-to-one, homologs, if you will, of the ones that were famous from Drosophila melanogaster. They published this paper in 2007, and it really is the inspiration for what we did. They looked at this one species, Drosophila ananase, which is about 15 million years diverged from melanogaster, and they found a completely different element. They didn't localize it to the chromosome end, but suggested the possibility that this one might also be a telomere specialized element. So what we've done in this paper is really fill in the phylogenetic space between melanogaster and its close relatives and this distant species, Drosophila ananase, with the hopes of being able to sort of date the birth and potential loss and turnover of these elements. And then in the process of, seeking to do a quite descriptive project, we came upon some surprises. So
just to jump in really quickly, so just these retrotransposon families have such interesting names. So these are the jockey elements.
It's the jockey elements, yes.
Yeah. Where'd that name come from?
That's a great question, Nels. I do not know.
Oh, I stumped you. Wow, this is great.
That's excellent.
I don't either.
That's a really great question.
But anyway, so looking at the jockeys, including the telomere, ones that were known to be associated with telomere, located out at the ends of the chromosomes, what did your phylogenetics start to tell you as you were filling in those additional species?
Right. So what we discovered was that there were sort of four distinct major lineages that we discovered across these different species. And these lineages, basically when we made a phylogenetic tree, as you might expect for a retrotransposon, didn't recapitulate the species tree. There was this discordance that when you map the identity of these elements back onto the species, what we observed was sort of turnover of these elements where, one species would have, for example, the TR2 element and maybe the TARA element, whereas its sister species would have neither of those elements and instead have this tartar element. And so there was almost this sort of engine underneath that we were seeing the products of that seemed to drive these elements to turn over time, which again, from a kind of selfish element perspective, is not surprising at all. But if it's an important mechanism of doing an essential function, it actually is quite surprising that you need to refresh effectively the transposons that are performing this ostensibly static activity.
So that sounds like it might be consistent with this, not symbiotic, where things are well behaved from an evolutionary standpoint, but more of a conflict scenario.
Yeah,
so can I ask?
Exactly right.
Yeah. So can I ask me? So kind of those 12 species that you mentioned that have really been a lot of resources and effort has been put into characterizing them. They're sort of now household names for evolutionary geneticists.
I'd like to.
But you have a few. Yeah. But you have a few sort of surprises, I guess, like Drosophila ropeloa. I've never heard of that species, and it sounds like it might have something interesting going on with its telomeric elements. What's happening there?
That's right. So Drosophila ropeloa was an unusual species, even from a phylogenetics perspective of the transposable element, in the sense that it had this lineage of retrotransposon that was clearly closely related to this jockey subclade, but it appeared to be distinct from all the rest. So it has an element called tartar, which was named by the lead author on the paper, Bastien, who's French, naturally. So tartar is sort of intermediate, effectively, between the tart element and the tare element. And, I wasn't even convinced that it was distinct based on the trees that we built. But when we went to validate this element as truly a telomere specialized element using fluorescent in situ hybridization on polytene chromosomes, those giant chromosomes that come from the salivary glands of fruit flies, we expected just to sort of check off the list, yes, this element is telomere specialized, as is every other element in this sort of subclade of jockey elements. And, to our surprise, these elements seem to localize not only to telomere ends, but also to other places in the genome, including the pericentromeric heterochromatin, which is something that we sort of speculated about in our 2017 paper looking at the evolution of the telomere proteins across these same species. And this innovation, or what it looked like a sort of innovation, an escape effectively from the end of the chromosome, was really exciting to us and sort of fit with our idea of conflict. And we infer that it's sort of an escape event as opposed to an ancestral promiscuous insertions that are capability which is then lost because all of the relatives of this element appear to be telomere specialized. So we infer this is an escape event, although, of course, adding more species to this tree would better support that inference.
Yeah, very cool. So you're saying that the tartar elements might have escaped from chromosomal jail that you were describing earlier. What are the usual mechanisms in place to keep these things sort of in jail, and how might have this one gotten out?
Well, that is our research program looking at the rapid evolution of the host proteins, which I know you're familiar with, Nels. The telomere proteins in Drosophila evolve extremely rapidly, including the elements that not only protect chromosome ends from fusing together, but also those that regulate the telomere length. And we sort of looked at this comprehensively a couple years ago and found indeed the host proteins that regulate telomere length. And when you knock out these genes, the telomeres grow. Possibly not surprisingly, evolve really rapidly under positive selection, so really enriched signatures of dynamic evolution. And so we speculate that the host proteins are constantly evolving to, in some species, certainly not all species, to keep these retrotransposons in their jail. And, maybe Ropaloa is an instance of, an event where the host lost. Now, we're able to sample this species today. So clearly it didn't lose too badly because it managed to be around for us to know of it and sample it. And I don't really have a good explanation for why that is, except that maybe, tartar escaped and, in just like any other retrotransposon, those instances where it, tartar inserted into a important protein coding gene, we don't sample those individuals anymore. We only sample the ones where the insertion events were not too deleterious. So we could sample it today.
Yeah. So that's a related question I wanted to ask you. And I should just say also, we might be running a bit short on time. So Mio is really generous to join us. She has a number of appointments today, but sort of fit in the episode of TWiEVO here. So first of all, real appreciative of that, Mia. But when you need to cut us loose, just don't hesitate to tell us. But
when you need to get out of jail,
that's right. You can escape at any moment you need to.
Nice, very nice.
But short of that, I think Vincent and I will just keep asking a million questions. So, and one I had related to what you just said is: so do you think that there are escapers like this that then sort of crash out the species? That there could be lots of cases like that, but we just don't see them because they've gone extinct?
That would be consistent with our model, but I hesitate to say anything too strongly in the absence of, hard data to support it. But yes.
So the other related question is, I guess if you compare the elements that are still in jail versus the ones that escaped, they're too different to be able to get any idea of what might be the causative changes.
Well, I don't think so. So the other part of our telomere research program is looking at these really fast evolving telomere proteins. And our approach so far has been to swap into melanogaster versions of these genes that encode telomere proteins, which are putatively naive to the retrotransposons that are maintaining chromosome ends in the contemporary melanogaster genome. And so we hope by doing these manipulations, we can actually detect, the particular proteins that might be engaged in sort of policing these elements and keeping them at the chromosome ends. We also have experiments going of actually putting in effectively the wrong species versions of the retrotransposons back into melanogaster and ask, in a naive genome to the retrotransposon, does the transposon actually start misbehaving? And so, this is really a kind of decades worth of work, but we're, starting to hopefully use these manipulations, get down to the sort of genetic determinants of these dynamics.
That's really. Those are cool experiments. I like that a lot.
Yeah, me too.
So these can I ask one more, Nels?
Of course.
I'm sorry.
Of course. No, not at all.
So they're basically two proteins encoded in these elements. There's a GAG and a reverse transcriptase, right?
That's right.
And the way they work is they reverse. Tell us how they jump out and get back in again.
That's another really great question that I don't have a great answer for. So, given the distribution of these elements at the chromosome ends, it appears that they get reverse transcribed at the ends of the chromosomes using the at the poly A tail, effectively the element at the very ends of the chromosomes. So they sort of show up in these head to tail arrays, and it's not totally clear whether these elements inherently have the capacity to actually insert other places. There's rare events of these elements throughout the genome, and what's a bit of a puzzle is that these elements, they encode what's called the endonuclease domain, which is in the second open reading frame that includes that reverse transcriptase. And many people have cited the strange observation that the endonuclease domain appears to be intact, and so the endonuclease domain would predict that these elements actually have the capacity to insert internally. But the sort of molecular mechanisms have really not been well studied up to this point. Yeah,
that would sound like the key element that, I guess that's like an integrase, right?
Exactly.
Yeah, that would be my bet.
Yes.
Go ahead now, sorry.
Oh, no, not at all. So I was just going to say that we've been talking about this one species where it looks like the tartar elements might have escaped, but you also found another species. This is Drosophila bioRxiv. I might be pronouncing that wrong, but something very different has happened with the telomere-associated transposable elements there.
Yeah, so this was, I would argue, the biggest surprise of the paper, again, having gone in just to sort of characterize the diversification of these elements, when Bastien came into my office one day and said, well, bioRxiv doesn't have any. What? We'll go back and find them, and he said, no, I found a jockey element that looks like, is related, but the jockey element is on the wrong side of the tree. The jockey element is where the sort of euchromatic jockeys are, not the telomeric ones. Mm-hmm. And so he made a fish probe to that jockey element because he figured, well, maybe that jockey element is the telomeric retrotransposon in bioRxiv. And in fact, it hybridized throughout the chromosome arms as we would have predicted based on its phylogenetic position. Mm. And so we took a deep breath and decided to subject this species to long-read sequencing using PacBio. And we expected to find maybe a non-jockey element that has been recruited and replaced this subfamily that appears to be in everybody else. And to our surprise, found no such sort of new recruit. And instead, what we found was a lot of satellite sequence of a particular class called SAR, so SAR and SAR2, the satellite sequence that is typically found, outside the telomere. And we also discovered these relics of a DNA transposon called helitron, which I had never heard of and quickly educated myself. It had shown up in other really interesting evolutionary genomic analyses, include Chris Ellison and Doris Bactrog. These helitron sequences have been shown to recruit dosage compensation machinery in neo-sex chromosomes. And so, we got pretty excited by what these helitrons might be doing. And in fact, because they're these relics, we really can't rule out the possibility that they're just the product of an ancient invasion event, possibly facilitated by the expulsion of these jockey elements, which are no longer there. But their inability to move around due to the loss of their mobilization machinery suggests that they actually are not functionally replacing the jockey elements that we see at other species' telomeres. So we think this species is a really surprising and striking example of the collapse of this telomere elongation mechanism. We speculate in the discussion section that this species is likely using an alternative mechanism, probably the one that yeast does and flies do and humans do, which is this recombination-based elongation mechanism.
Yeah, so it's really wild, just all these dynamics, both the gain and in this case, it looks like the complete loss. So just maybe, and again, I know we're short on time, but so do you think there's sort of a picture emerging here or clues about kind of the origins of telomerase? Like it just seems like with all of these elements possibly competing against each other, also with the conflict with the host, as you're describing it, how is it that telomerase, which probably emerged, I guess, from one of these sort of mechanisms with the reverse transcriptase activity, how did that sort of come to dominate a lot of eukaryotes?
So that's a very deep evolutionary question that I'm quite uncomfortable speculating too broadly about. So it's great that we're short on time, so I can escape.
Right. You can always can give us the slip.
We do know that the reverse transcriptase of telomerase, if my memory serves me, is related to a different class of retrotransposon called Penelope elements. Whether retrotransposons represent an ancestral state deep in the eukaryotic tree, and telomerase was an innovation that then really stuck because it's a very good solution and resolves any potential conflict that you would have otherwise. I really don't know, but it is quite tempting to speculate that telomerase did solve a problem. I think I'm going to leave it at that.
All right. Mia, thank you so much for joining us.
Thank you so much for having me. It's been really fun.
Yeah, thanks, Mia. Great to hear from you.
All right, appreciate it. Thanks.
Bye. So let me get this straight now. So this is totally new to me that flies, Drosophila doesn't have telomerase. There's not even a telomerase gene, apparently.
Yeah, all completely gone. No sign of it.
And they probably had it at one time, right? Because telomerase is old, right?
Exact. Yep.
And now it uses transposable elements to do this. I think the idea that sometimes they stay in the jail is really cool, and that's a great mechanism that she's looking at. But and it probably got out of jail now and then. But this last thing, this bioRxiv, they don't have telomerase, and they don't have transposable elements in the telomeres, right?
Yeah, exactly. So then now it's the closest. It's really wild, and as Mia was sort of saying, gets to this alternate mechanism based on recombination, which comes in and is, I think, gaining more attention. As that's been observed and associated with cancer-related states for cells when they kind of go off the rails that way. And so that's still. So there's that other kind of alternative mechanism, and yet. Telomerase still persists sort of far and wide from an evolutionary view. So it's really, I think, just fascinating how many different ways there are to sort of, so I guess maybe the one common thing is that if you have linear chromosomes, you have to deal with that erosion issue, the end replication problem.
Yeah.
And so there are just all these sort of stopgap measures. And then when you overlay that with all this volatility that Mia is discovering and describing among the Drosophila species, really interesting biology, and kind of illustrates how something, at least for me, when I was, as I was kind of mentioning at the top, when I was learning about these mechanisms originally, something that's totally critical, vital to all cells as they're dividing, and yet the fact that there could be all this underlying flexibility or conflict or volatility is really surprising and exciting. Yeah.
I thought telomerase was a good solution, although it did seem a little hacky, right? It's a bit of a hack. But
that's right.
But I take the virus perspective, right? And there are really interesting solutions to the end problem in viral DNA genomes, right? So some are circular, so there's no end problem, right?
Yeah, that's right.
Some are linear, but when they get in the nucleus, they circularize. So that takes care of that. And others utilize a protein primer, not an RNA primer, for the first priming event. So there's no loss of RNA sequence, which is what gives you that end issue to begin with. So isn't it interesting that viral genomes don't have this issue? They don't use telomerases, as far as we know, although there might be some out there that do. And I just wonder why don't cell genomes use protein primers? Why don't they circularize and get around it, right?
Yeah, I see your point. So that would be a more kind of, if you were a genetic engineer back in the origins of eukaryotic cells, this is what you might imagine. Well, or more recently, something could kind of, the same way that these TEs are sort of competing and taking over or propagating, that something like this, something more elegant.
It didn't work. I suspect that circularizing a really big chromosome is no good for some reason, right? It probably breaks or whatever. And there are obviously reasons. So this transposable element end of chromosome solution is relatively recent, right? In terms of, it's 40, 30 million years old or something like that? Is that the...
Yeah, well, might go, so I'm trying to remember now. So there are some, as Mio was mentioning, there are some other papers just doing a kind of a broader phylogenetic, treatment of that strategy. So certainly independent events, in the moths, in the crustaceans, for example, where other families of TEs have come in. But, so that could put it back certainly deeper in evolutionary time, sort of an independent events. But yeah, for this Drosophila case, that this in the family of these the sort of subset of these jockey elements in particular that Mia's been characterizing, that's much more recent. That's a pretty recent kind of innovation in a sense, or development, I guess, for the. I mean, but the point that you made, which is still quite interesting, is the fact that you have no sign of telomerase at all, and so. If that was just kind of happened yesterday, you would say, oh, there's probably like a stop codon or a pseudogene kind of remnant of telomerase, and yet it is vanished completely. And so that sort of is an interesting clue about how fast that sort of turned over, how long it's been, so that there's not even sort of a trace of it left behind.
Yeah. So could species that have telomerase change to use transposable elements?
Yeah, interesting question. I mean, so there, of course, now, that alternate mechanism with recombination in place, but I think you could imagine some fun experiments, and maybe, sort of high-risk experiments of high-risk meaning that they would actually work, but to try to engineer sort of that transition. And you could imagine, even thinking a little bit about, could you sort of hobble, like use some gene editing or something in a mammalian cell, and hobble telomerase to some degree, while at the same time offering kind of a Drosophila-like telomere-associated transposable element. That'd be pretty cool to try.
Yeah, you could put a TE near the end of a chromosome, right? And just set it up so it doesn't have to find it. Hobble the telomerase, as you say. I guess I don't know what you would do it in, because I don't know if mouse cells in culture even produce telomerase. I don't know. Maybe they do, and that's why they can be immortal, right?
Correct. So and Mio, I think, was hinting at that as well. So the difference between sort of the germline, somatic, and then also when you're transformed, and that's when telomerase activity can return, and that's the case, I think, for a lot of these transformed lines. In fact, some primary lines are immortalized by
putting
overexpressing telomerase. Yeah. So, the real trick here, and this gets at a paper that Mia's group published, I think, a couple of years ago now in MBE, Molecular Biology and Evolution. And she spoke a little bit about this, is, this whole collection of host genes that encode functions, that sort of the jailers or the police personnel, as she was using in that sort of interesting metaphor, the kind of collection of genes is different than a lot of other eukaryotes that actually have telomerase. There's some overlapping functions for sure that are involved, but then telomerase is gone, and there are other genes encoding proteins that now sort of have taken on these new functions. And so that would become a question, I think, for this kind of experiment that we're kind of daydreaming about on the mammalian side is how many of those factors do you need? How did they kind of come up to, as part of that kind of almost genetic conversation between host and selfish gene? Really fascinating.
So I would take a mouse cell line, an immortalized mouse cell line, and just decide which transposable element, maybe one that already exists in the mouse genome, or maybe one that doesn't exist so that it's not restricted in any sense. Put it in, give a few generations so that the thing can transpose, and then knock down telomerase with siRNA or shRNA, something like that, and see if the cells grow past, 20 or 30 more divisions.
Yeah.
Is that sort of the idea?
What could possibly go wrong?
Well, lots could go wrong, but
that's right.
it's not a horribly complicated experiment, right?
It's not. So I think, what might happen is the trickiness of that would be, okay, well, what if it didn't work? Then how could you gain, you could imagine, I think, many ways that it would fail, and then the cells would just sort of stop dividing. So could you somehow include some selection in the scheme? So that if the transposable elements somehow were engineered so that they also encoded some kind of marker that you could select on, so that you were, that might be something to think about, and then the hope would be that you could do millions of experiments with some sort of, variation cooked in or some collection of different treatments, and then select back and just gain a foothold on a marker. But I have to tell you, Vincent, I think it sounds really exciting. I also think it sounds like a really, evolution in all of the natural experiments that have been done over millions of years, hundreds of millions of years, and continue to be done today, just in natural populations, all the biodiversity around us. In some sense, it's amazing how telomerase has held on for so long, in that
clearly
these are more correct. Yeah, these are more the exceptions than the rules. And so, what were the sort of set of unlikely events that all came together to sort of tip the balance? I think it's really exciting to think about.
I'm not even sure that a mouse would be the right organism to do it in, but, one could think about this quite a while. And this is kind of the experiment you wouldn't want to put someone on whose career depended on it. Right?
That's right.
Like you or I could tinker and do it now.
Now we're talking. Yeah. Yep. Well, and I think as Mio is also describing, sort of these experiments the other way around, with using all of the great genetic tools available in Drosophila to start to manipulate that system, as she was saying, like swap in different ones, potentially.
Yeah, that's very cool. That's very cool. I like that a lot. Yeah,
I agree.
I think that's going to work because they have both kinds of telomeres there. They have both kinds of TEs, ones that stay in telomeres, ones that don't, and they can put one in, so they could take a line of melanogaster where the TEs are in the telomeres, they stay there, right? They could introduce one from that other strain that has moved elsewhere, right? And see what it does in Drosophila. Is it restricted or does it escape, right?
Yeah. Yeah, it'll be fun to see. Cool. I think, as she said, they have lots to do over many years. I bet there'll be some. Cool. I agree.
The other thing I wanted to mention, you talked about, you used this really cool analogy that it's weird that cells have turned over the keys for this essential end of chromosome function to transposable elements, right? The idea, because these are foreign. They came from somewhere else, right? They're not, well, now they're part of the genome, but they originally are parasites, right?
Yeah. So this is emerging in not just the telomeres, but other cases where what would ostensibly just be a selfish genetic element that just over-replicates, and that's why it exists.
Right.
But that hosts on kind of rare occasions, but we're given enough evolutionary time again, we're starting to see and uncover more and more of these rare events where you see kind of the remnants, both of transposable elements, but even like endogenous retroviruses as well. So like one of my favorites are the in the placental development. So these syncytin genes, right? I can't remember if these have come up on TWiEVO yet, but
totally, And your interferon promoter.
Yeah, right. Yeah, exactly. So that we did a study ourselves looking at from these LTRs, the kind of terminal repeat regions that get potentially reclaimed for their ability to act as promoters, and then rewire, in essence, our immune responses for sort of the challenges ahead.
And she calls this domestication of the transposable element. In other words, you let it stay in the genome at one place. You don't let it move around, because that's when bad things can happen, when it can go stick into another gene or a different part of the chromosome, and it's deleterious. So that's the interesting part, the domestication, right? How do you get these things to stay? And that's what the experiments that she was talking about. But this is all going to be relevant in our next episode, because I don't know how far you've gotten into the book by David Quammen, but he talks a lot about phylogenetic trees and how you can't just pay attention to the organism's genome, because there's so much horizontal gene transfer from others. How do you draw the tree? And he has I just finished reading last night. He went and talked to your buddy Cedric Fichot when he was over in Utah. You probably missed him.
I think I was out for lunch or something. I missed that.
I talked to him about, he was among the first to start looking at these transposable elements, and when the first genome sequence came out, and then he went to the guy in Paris who discovered the syncytians, and the domestication. So he's really into that whole idea. So we'll have a good chat with him.
Yeah, great preview for TWiEVO 37. Stay tuned for this one, everyone. So I think both Vincent and I are really excited to have David Quammen as our guest, who just published this provocative book, The Tangled Tree, that gets at exactly these some of these issues. Thinking about vertical inheritance and some of the rule breakers, which are turning out to be really interesting.
Yeah, it should be fun. Yeah.
Yeah, agree.
Did you have anything else on this paper that you want to chat about?
Yeah, that just about covers it. So there was, a few details, if Mia could have stuck around, that I would have sort of tried to drill down on a little bit. But check out that preprint on bioRxiv, really interesting. And a nice example, again, of how having kind of at our fingertips the ability to do really in-depth genome scale analysis. So she mentioned using PacBio long reads to, they found that interesting case where they said, hey, wait a minute, it looks like it's missing, and then could actually kind of prove that by doing a quick and comprehensive genome-wide view using this new technology. It's another example, I think, of the possibilities that are just opening up based on the combination of evolutionary thinking, the tools in a system like Drosophila, but also the technology that's coming along as well. A really fun time to be doing this kind of work.
Yep. Well, Nels, we have one email.
Oh, great.
And why don't you take that one, because it kind of involves you.
Sounds good. So Mike writes, hello, I am a PhD student in Paul Turner's lab at Yale and enjoy listening to your podcast. Recently, I was listening to one of the older episodes, and you were discussing scientific publishing and open access. I was wondering if you're familiar with and how you feel about the peer community in evolutionary biology, the PCI, Evo Bio, concept. I was introduced to this idea at the Evolution Conference in August, where I also had a chance to meet Nels after his talk. Basically, the idea is that they can serve as a community-driven peer review process for preprints, which can supplement or replace journal-focused reviews. They have a good schematic on their website, and Mike includes a link that we'll put on the TWiEVO notes to check that out. Anyway, I really like the idea and think it has a lot of potential to allow open publishing and possibly a move away from for-profit journals, and was wondering what you thought of it. Thanks. Well, Mike, first, thank you for writing in. And I remember talking briefly at the Evolution Conference in Montpellier. It was a really fun event. We were in a session on virus evolution. And so, Vincent, have you looked at this schematic that Mike pointed us to?
Yeah, I did, and I really think this is something I have been thinking about because, on almost every podcast I do now, I complain about publishing in some way. Mm-hmm. And the idea is that I don't like that the journals are controlling our publishing and our career, right? And this is so a couple of weeks ago, I gave a talk at Sinai here in New York, and I talked about the publishing problem. I said, the ideal solution would be to have science be journal agnostic. In other words, you just publish it somewhere, it's peer reviewed, and people just like it or not due to the work, not where it's published. Because it's not good for our jobs, our grants, our promotions to depend on where you publish. It's crazy, right?
Yeah.
So I thought, well, bioRxiv peer review combination would be great. And that's exactly what this is. Here you put your manuscript on bioRxiv like the one we talked about today.
Considered.
And then you submit it to this PCI, and they peer review it, and it gets accepted. And then on bioRxiv, it's an accepted manuscript. You don't need a journal. I think this would be great. One place for everything, right?
Yeah. So I was, that's right. So I was at a meeting. I can't remember if we discussed this, but at this last winter, it was at the Howard Hughes, and it was in collaboration with this organization, ASAP Bio.
Right, right.
And a similar conversation emerged, actually, in some experiments that are being done now at the journal eLife, sort of along these lines. And so just to kind of also just so in the schematic, what it shows is, as you were saying, you put on the bioRxiv, and then you would submit it to the PCI, this peer community. And then so in some sense, they are acting a bit like a journal. So they're considering whether or not they will put it out for peer review, if they'll try to recruit reviewers. If they make that decision, then it goes through that. It could still be quote unquote rejected through the so-called PCI process, or recommended with, I'm guessing, and this was a topic of that HHMI meeting, with a transparent view of what the reviewer said and sort of how that process played out. I think what's a little different here than about, sort of the journal model is that instead of going into a single journal, what happens is that recommended preprint, if it made it positively through peer review in the PCI, so-called PCI process, then it goes back, or it's still on one of the preprint servers, the bioRxiv, the archive are related. There's a few new ones, I think, that are just starting to pop up as well. But it has sort of a tag, or this sort of seal of the PCI process. And I'm guessing those peer reviews, some summary of them, similar to the eLife model. But then, they do note that then at that point, you might take your peer-reviewed preprint and shop it out, as they say, so it becomes a valid and citable final article, and you can still be submitted to a journal. So, I think, so first of all, I totally agree with you, Vincent, that the amount of sort of time, effort, even, the sort of incentives as people are weighing promotion and sort of making it through a career, getting the attention of, job application pools and things like this is totally out of balance. But then, I think we're still kind of tinkering and figuring out the way forward. I can imagine on the other side, for example, with this, if you've gone through a peer review process and then send it out to a journal for another peer review process, think about the peer reviewers. This is a lot of work. We're now doing double peer review. Yeah.
I think the journal has to take the reviews that they have already and decide what to do with it. But personally, I would cut the journals out completely. I can see this. This isn't perfect, but I'm glad that people are thinking of alternatives, right?
Oh,
and
yeah, definitely.
I could see something like this or a modified form growing, and eventually the dependence on other journals would decrease.
Mm-hmm, mm-hmm. Yeah, no, I 100% agree. And I really like this idea of experimenting with different pipelines, different ways,
right.
And then seeing kind of what works, what doesn't work, maybe refine, and then see if something picks up.
Totally. I don't think we're going to get the solution right off the bat.
That's right.
But I think this is better. And the one thing I don't know much about here, they have this part where you submit your paper to PCI, and then there's a little arrow. It says not considered for peer review. So, see, that's the problem with the journals right now, is that they say, we're not interested in this. So this would have to be better than that, where they would say, well, this is interesting, but you haven't done this critical thing or whatever. Not just reject 80% of the submissions like the big journals do, maybe 5% for whatever reason, right? Because I think most papers should be peer reviewed. That's the whole problem we have with these other journals.
Yeah, that's right. And then, but you can't at any point along here, you kind of shift the burden to sort of a step along the way. So doing, if every single thing that came out was peer reviewed, that's not a small amount of work. And so
sure.
So that triaging that, or determining sort of which ones do and don't, that issue won't go away regardless of how we sort of organize the dissemination of these papers and so of these manuscripts. I mean, for me, the first major breakthrough that is clear and is part of this pipeline, part of what you and I have been doing today, talking to Mia, is that because now the bioRxiv and other preprint servers have come sort of to be adopted, this has just been so great in terms of getting the information out in a very timely way. And so that to me feels great. Then the next step's a doozy. It becomes tricky to figure out, okay, just given the time constraints for peer reviewers, who's deciding what gets peer reviewed or not? That's still something that we're working on. And I think, my hope, and I think optimistically, that as eLife is experimenting, as PCI, this process that Mike brought to our attention, as other things are crop up, and maybe some that we don't even know about yet that might gain some attention. It's clear that we're putting our finger as a community on a real problem here.
Now, just one more thing here. Mm-hmm. Society journals, ASM, Microbiology Society, they review most of the papers that get sent to most of the journals.
That's
so
right.
Journal of Virology, I know, I used to be an editor. We reviewed almost everything that was submitted. We had a lot of submissions. It can be done.
Yeah.
There are a fraction that clearly don't get reviewed, but, Nature reviews 15% of what's sent to them.
Yeah.
That's ridiculous. It creates an artificial exclusivity, and that is not good for science. That's the whole thing. And even eLife doesn't review everything. I don't know what, they'll say, we're not interested in this. How do you know? That's the thing that drives me nuts. How do you know that this is not interesting?
They've said it to me a few times. I can tell you that.
Yeah, it just happened to me last week. I had an editorial rejection from a journal, and it just makes me mad because I know it should be reviewed. Right, let the reviewers decide. And meanwhile, the journal that I submitted it to and was editorially rejected is publishing stuff which I think is not as good. So, I think that this has to change. And I love this idea. I'm glad it's evolving. And thanks, Mike, for sending this because I wasn't aware of this. And I think it's great that people are thinking of ways to get around these big journals.
Yeah, agree.
Cool. All right.
Yeah.
How about some picks, Nels?
Great idea. Science picks of the week. So I've got one I'm really excited about here. This is a new podcast, a new science podcast called Cinema Science, and a great website. I'll put up the link for our listeners. And just so their tagline is, scientists talk about their favorite movies, games, and TV shows in this podcast. Every other week, Anne and Heidi interview a researcher about their favorite piece of media and the science behind it. So I know a little bit about this effort. So Heidi and Anne are two neuroscience graduate students here at the University of Utah, and they've created this new Cinema Science podcast. They've already got a number of episodes together. They've really put some hard work into this. They convinced their program to do some financial backing and used that to sort of set up their own podcasting studio. And they already have some interesting episodes. I'll use my tagline, spicy episodes online here. So do you remember that movie Gattaca?
I do.
From a year? Yeah. So they have an episode on that. Designer baby astronauts editing the murder gene. Also some Jackie Chan movies, The Drunken Master. What is the optimal headbutting technique? I don't know how much science is behind that, but it certainly sounds intriguing. Alien versus Predator versus Space Cows, the 5 alien senses. It sounds interesting to me. And a number of other episodes where so basically they're taking their friends, colleagues, or other scientists, and as the tagline said, kind of thinking about the science behind the movies, games, or TV shows that sort of capture the attention or interest of their guests. So this is available now. It's up on iTunes, Spotify, and Stitcher. So check out Heidi and Anne's new podcast, Cinema Science.
That's pretty cool. Good for them for getting, support locally.
Yeah, definitely. And just to say, so I've kind of watched, kind of just because I'm in the neighborhood here, and it just reminded me, seeing them develop their podcast, how lucky I am actually to be a part of this podcast. So as they have been figuring out what does it take to set up and run a podcast, I just sort of cruised in here and have relied very heavily on all of your expertise over the years at the TWiEVO in the Twix empire to just sort of bootstrap up to this level of podcasting. So I've been really lucky, and I give Heidi and Ann a lot of credit for building theirs from the ground up.
Oh yeah, that's the way I want to do it. I want to take what I learned and get other people like you involved. Sure, absolutely.
I really appreciate it. Yeah,
you can't do it all yourself, you know.
That's right.
So cool. That's great. Good luck to them. Hope they keep going.
Yeah, exactly. Yep. How about you, Vincent? What's your science pick of the week?
So I was looking through. I like Instagram a lot, and I post a lot of pictures there about sciencey stuff, and came across one the other day, which was an amazing. This is a picture of a mastodon being unearthed in Michigan. Huh. And it was this amazing picture of a big hole, and a crane was pulling out the skull still attached to the two tusks, right?
Wow.
And it said something like this had been slaughtered by hunters years ago. And all these people were standing around in the pit and around it, and the thing looked just huge. And I said, I have to find this. And it turns out it was from an article in some archaeology magazine. And of course, when I'm putting the pic in the show notes, I can't find the original Instagram picture because that's the way it is. They go away. And so I just Googled it, and I found that, in fact, just back in 2017, some mastodon bones were dug up at a Michigan construction site. So I found an article in a local newspaper, Grand Rapids News, mastodon bones unearthed at Michigan construction site. So they have a bunch of pictures, and I have a YouTube movie showing the thing being pulled out. So it's just that the skull and the tusks are all still connected, which is really, really amazing. And then I found a link to another article where they're putting the bones together at a local museum. Mastodon skeleton reassembly gets underway at the University of Michigan. So this is from the Museum of Natural History at the U Michigan.
Oh, wow.
And so they cleaned it all up, and it's 11,000 years old. And,
very cool. So, yeah, just hiding in plain sight. So it looks like I'm looking at your links here, and they've got it. There's like an old Phillips 66 sign next to the jawbone or something like that. Kind of crazy or maybe a reminder to keep your eyes open, right, when you're digging, or there's all kinds of amazing stuff right under our nose.
No, it's very cool. And actually, the Michigan thing may not be the same skeleton. I don't know. But the one article in the Grand Rapids News is definitely the one I saw on Instagram, because they have a little video down at the bottom that you can play, and you can see the video preview is exactly the picture. It's amazing that it's intact, right?
Yeah, that's right.
So I thought it was cool.
Well, I agree. And then the next question is there any DNA preserved in that old beast?
I bet there is. It's 11,000 years. It's not too bad. And of course, it's going to be contaminating DNA, right? I guess you could look inside the bones, right? You could go look in what used to be the marrow, I suppose. See what you can get. Anyway, so
another emerging and exciting area of ancient DNA that we've touched on a little bit on TWiEVO, and I expect that we'll be talking about it more as a lot of advances on that front too.
Bet.
Yeah, cool pick. Yeah.
All right, that's TWiEVO number 36. If you want to send us a pic, you can do that. You can just email it to TWiEVO@microbe.tv, or you can send us questions and comments. If you listen, you're probably doing it on a cell phone or a tablet. You're using some kind of an app. Please subscribe so you get every episode. That gets us more subscribers, so it looks good for our numbers. And if you use a Play program that lets you rate podcasts, like give it a star or something, please rate us. That helps us send us to the top of lists so more people will subscribe, and that would be great.
That would be great.
And of course, if you really like what we do, consider subscribing or supporting us. And in fact, you'll be supporting all the podcasts in Microbe TV, which includes TWiEVO, TWiV, TWIM, TWIP, Immune. You can go over to microbe.tv/contribute. We have a number of ways that you can do that. We'd really appreciate it.
Definitely. Be grateful for all the support we can get.
Nels Elde is at cellvolution.org. He's on Twitter as L Earlybird. Thanks, Nels.
Thank you, Vincent. This was fun. Glad we could catch Mia for a few minutes and hear about her exciting work.
Yeah, there's nothing like getting the story from the people who did it, right? Who did the work.
Agree.
Mia Levine is at the University of Pennsylvania in the...
She's in the Department of Biology, and then I think also associated with the Epigenetics Institute, as you were saying as well.
Epigenetics Institute. And we'll put a link to her website so you can check out the other things that are going on. We appreciate her joining us. And I'm Vincent Racaniello. You can find me at virology.ws. Music you hear on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. Been listening to This Week in Evolution, a podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.